Hi, this is Andrew Bacon, and I am in your 7th period biology class in the 2021 and 2022 class year, and I'm going to teach you how mitosis and meiosis works.
This is my mitosis and meiosis project. Mitosis is a process by which a cell replicates, creating two identical daughter cells. This will happen 10 quadrillion times in your lifetime, and that is how big the number is. If it helps you remember, it is a huge amount of times it happens in, and it's like unreal to even like imagine. This is the all the steps of mitosis, and its first step is interphase, which is kind of an umbrella term for the whole entire thing. But then prophase is the second, the third is metaphase, the fourth is anaphase, and the fifth is telophase. And sometimes you'll hear cytokinesis as the end, but really that's like happens in 1 second, and it's the splitting of the two cells, which you can kind of see in 5. So that's the 5 main steps, but you may hear 6 as cytokinesis, which just gets clumped in a telophase usually. So the first phase is interphase, which of mitosis, and it's just a beginning process. Nothing's really happening. There's not a whole lot going on. So prophase is the second phase of mitosis, and where action starts to happen. The chromatin starts to condense, and the nuclear envelope breaks down. And when this breaks down, the chromatin and chromosomes can start to float around, which leads to metaphase. And in metaphase, the chromosomes start to line up in the middle of the cell. M for meta stands for the middle. It's a way to remember it. And the spindle fibers start to attach to the chromosomes, which you can see in the picture. And they call it the metaphase plate, which is an imaginary line. It's just really the middle of the cell. And anaphase, the chromosomes break off into sister chromatids, which start to pull off into opposite poles/directions. The prefix ana means back, and they kind of move to the back of the cell, opposite poles. So that helps you remember. And the chromosomes break into sister chromatids, which has the same DNA, and this is very important to remember when you have the two different identical cells, because these DNA are the exact same. And this is telophase. In telophase, the one cell starts to divide into two cells, seen in the picture. The chromosomes are divided up into two different identical cells, and they start to create new organelles in other parts of the cell. So the nuclear envelope reforms, and the nucleus and other organelles start to form. The centromere forms for future mitosis processes for the cells, and the cleavage furrow breaks, and that's when cytokinesis happens. And yeah, cytokinesis is the official splitting of the two cells/cytoplasm, making two identical cells. So this is it's instant, it happens right after. So mitosis is now complete after cytokinesis. So now we have two identical daughter cells that's the exact same. Now this is meiosis, which mitosis and meiosis sound very similar, but they're very different things. The process of meiosis creates 4 different daughter cells, each with half of the genetic material. This creates sperm or egg, depending on if you're male or female, and that have the genetic material. During reproduction, these two cells merge together to form a cell with 46 complete chromosomes, or 23 pairs of chromosomes. This is the really long cycle of meiosis. One way to remember meiosis is that it's like mitosis happening twice, which may confuse you, but that helps remember it like that. And you may have to turn your head a little bit, but you have interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis, prophase 2, metaphase 2, anaphase 2, and telophase 2, and cytokinesis. So you see how it's happening twice. You have the 1, and then it splits, and then 2, and then it splits again. And you can see the 4, which they call granddaughter cells. Now, meiosis 1, prophase 1. Prophase 1 in meiosis is where it all starts. The spindle fibers are being created, and the homologous chromosomes pair up. So it's like starting everything. Metaphase 1 of meiosis. Like in mitosis, the homologous pairs align to the center of the cell, and the spindle fibers attached to the centromeres. So that imaginary line, the metaphase plate, is being played here, and the metaphase plate and the spindle fibers are attached to the centromere, which are a part of the homologous chromosomes. So note, the chromosomes are identical right now. In anaphase, in anaphase 1, the homologous chromosomes start to move to opposite poles/sides of the cell. This is that ana back, so these homologous chromosomes are being pulled to the back of the cell, opposite sides. And then in telophase 1, in meiosis, the cell starts to split into 2 different cells. And then right after telophase, we all know cytokinesis happens. So now we have 2 identical cells, but it needs to happen again. So meiosis 2, prophase 2. In prophase 2, the chromosomes condense and DNA does not replicate, because that would make too many copies. It would mess things up, and you'd get like mutations and stuff, which we don't want. Metaphase 2 of meiosis. In metaphase 2, the chromosomes line up in the middle in the cells and start to split, and the spindle fibers attached to the centromere. So this is very similar to the other stuff, but see how they're different now? They're the same, but they're different. They're not homologous. Anaphase 2 is when the chromosomes go to the opposite ends of the cell. So like we already knew this, the centromere divide, chromatids move to opposite sides of the poles. So now we're starting to get our 4 different cells. In telophase 2, the chromosomes gather into nuclei and the cells divide. In telophase 2, the 2 cells divide, and now there are 4 different cells, which could be sperm, egg, or whatever. It's depending on your male or female. If you're male, you'll have sperm, and if you're female, you will have eggs. And this in meiosis is only for sex-related cells. In mitosis is only for body cells. So meiosis is now complete. This is another diagram you can relate to. I hope you learned how mitosis and meiosis works now. Remember, mitosis makes 2 identical daughter cells, body cells, and meiosis makes 4 different cells, sperm or egg. I am now going to show you a video of mitosis being shown in animation form, which may help you understand it more.
Interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has 2 sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into 2 sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus.